Welcome everyone. I'm really pleased to be able to give this presentation today about enhancing transparency in research with preregistration based on example of my ERP study. And I would like to ask you, before we dive into the topic, how many of you have seen perhaps before some nice brain imaging pictures, figures in different publications? So I'm not sure whether all of you are reading this kind of papers, but perhaps also in popular science blogs or publications, you may also have came across this kind of figures. But perhaps not all of you have thought, okay, how researchers may present this kind of figures based on their raw data that they collect. So here we can see two examples from a fMRI study on memory, and also a graph based on the ERP study from my own research. And in all the neuroimaging studies, neuroimaging data are very complex. For fMRI research, we are talking about here very precise spatial resolution with cubical millimeters precision. And in ERP studies, we can talk about very high temporal resolution, because the data are expressed in milliseconds precision. So beyond those, this sensitivity of the data, the amount of data that could be collected from just one participant is really huge. So usually the normal, I would say, raw data sets from a participant is over 1 gigabyte, of course, depending on the length of the time of the recording. And this big amount of data makes researchers we need to pre-process the data as researchers in order to be able to analyze them and produce any nice figures, and also to interpret the data in a statistical way. And this pre-processing process could be quite complex and quite difficult. So it involves many steps. Some steps are, however, more standardized, because you may not be able to do one step before doing the step before. However, there are many subjective decisions that a researcher may need to take in order to for example, say, to make a decision who has to, which data to exclude or which to include. So how to define the outliers, or how to define the artifacts in your data. So those could be really sensitive decisions to some subjectivity of the researchers. So here I just displayed some general data pre-processing pipeline for fMRI research. However, in real life, it could be much more complex, of course, depending on the study. So here is just another pre-processing pipeline, which already displays a little bit more steps. And also in my research, EEG research, I remember that the pre-processing pipeline that I created, it was about 20 pages, not only of coding, but in general of some of the steps and the description of the steps. And when I was trying to minimalize it, I think it took me like a little bit more than 5 pages A4. So it's really complex. And this complexity creates another problem. Well, it could lead to flexibility in pre-processing, and analysis of neuroimaging data. And recently there was a research published on exactly on this topic, which illustrates the problem. And it involved the team of over 70 independent teams who analyzed exactly the same fMRI data sets and tested exactly the same hypothesis. And maybe you would guess how many of those teams have applied the same analytical workflow and found the same results. So what would be your guess? Maybe 1, 2? Yeah, I see Sander shows exactly 0. So unfortunately, no two teams chose exactly the same workflow to analyze their data, and this flexibility resulted in differences in the results. And in general, this analytical flexibility could limit the conclusions that we can make based on different analytical workflows. So we have to beware of this, not only when we read neuroscientific studies, but also we have to be aware of our analytical decisions that we make, that how they may affect our own data, our own results. And a possible solution to little bit limit this problem could be, for example, validating and sharing complex analytical workflows. So it could involve that I, for example, would conduct my study, and after I conduct my study, I share the protocol and code so anyone could reproduce it. However, it should be also the different people should perform multiple reports on the same data sets in order to check whether there is any possible flexibility possible, or perhaps the data, the workflow that I shared is good enough so anyone can reproduce it. But in general, this could be, the flexibility could be also avoided by using and developing different analytical tools, which are based on algorithm. So there are, for example, different preprocessing pipelines available that are more automatized. It's available for fMRI research, but also for EEG research. So depending on what you need, you may try to look for different automatized preprocessing pipelines. And finally, the different analytical approaches could be also combined in meta-analysis to exactly see how different approaches affected the outcomes. But the last thing that you can do is just to be transparent, and actually before you do your research, you pre-register how you exactly want to do it and how you want to pre-process your data. So this would be the best solution for our research. And how to do it? So I would like to answer a few questions. So first of all, what is the pre-registration? So pre-registration could be understood as a time-stamped and available online plan, which should be accessible at least upon publication, which could involve hypothesis, the data collection plan, the pre-processing steps, and the statistical analysis plan. And why it could be important to do it? Well, first of all, it would, of course, increase transparency in the research, and it could limit the analytical flexibility that I was just discussing. But it could also limit two types of biases. The first is the confirmation bias. So researchers may be searching, interpreting, and remembering information that could support their prior beliefs and while ignoring the evidence against them. So some researchers may make it more intentionally, but some of them may make it more not intentionally. So anyway, we are humans, so let's try to limit this confirmation bias. And the other bias is the hindsight bias, which refers to the tendency to overestimate the extent to which the past events were able to predict present outcome. So for those two reasons, we should use the preregistration. And moreover, preregistration can enhance the good planning of the research. So you can improve your design before data collection starts. So, for example, when I was pre-registering my study, it really made me think through everything. And I could still improve some parts of my design, perhaps limit one factor which was not necessary to make my design more straightforward in order to make my study better. And finally, it could save time after the data are collected. So also this is from my own experience. When I conducted my pre-registered study, I immediately knew what I have to do next. And I just was following the pre-processing steps that I prepared before, or also the things that I said in my pre-registration that I plan to do. So it's really like a guideline for a researcher, and it saves a lot of time, and also it's just a very, very good tool to apply. Okay, how to pre-register. So there are 3 general rules of the pre-registration. So first of all, pre-registration should be specific. So you need to include the descriptions of different phases of the research workflow, from the initial design of the study to the final manuscript. So anyone could actually reproduce your findings if somebody would like to. It should be also precise, and it should be interpretable in only one way. So there should not be able to interpret it in the other way than it's stated. It should be also exhaustive in a way that, well, it can also, it should only be diagnostic to confirm or falsify the predictions. And you should also make a clear distinction between primary and secondary analysis. So the analysis to confirm your hypothesis, and perhaps some additional exploratory analysis. And the final suggestion is that it's very wise to run a pilot study before registration. By doing this, you can check whether your recruitment is feasible, whether your randomization works, whether all the procedures and assessments, measurements works. You can make sure that the instructions are clear for all the participants, and of course you can validate your task or the stimuli that you want to use. For the ERP research that I was intended to do, I could confirm this way whether the target ERP components were elicited, and also I can check whether the pre-processing pipeline did not include any bugs, some errors, and whether it's feasible to do it the way that I planned to. And another note is that the pilot tests should, well, usually people are doing pilot tests on smaller samples, but if you do it, then you cannot really use the pilot test for an a priori power analysis to estimate the needed sample size. However, if you use big enough sample size for a pilot test, then you may try to estimate your a priori power analysis based on the pilot. So in general, try to use bigger sample size for the pilot test. And okay, the final thing where you can register. So there are different platforms where you can register your study. The more detailed registration could be done via Open Science Framework. So you can register your preprocessing pipeline, and also you can write a pre-registered report, which is more extensive than just pre-registration at aspredicted.org. However, if it's your very first study that you're registering, I could suggest you to first try as predicted, because it includes only 8 key questions, and usually it's very short and straightforward registration. But still, it makes the registration valid, but does not allow to register, I think, more than around 2 A4 pages. So you have to be very concise. Now I would like to share some insights from my registration of ERP study. So I registered my study as predicted. It's available online for now in a blind version, because I'm still writing a manuscript based on that, and I would like to use the registration in the manuscript. And I also did a pilot study for the current registered study, and it was actually it was just published in Psychology of Popular Media, and I also included here preprints. So after if you're interested, you can also have a look at it to compare the registration and my pilot study. And some small background information about the study. So my research is about violent video games and how they may affect brain reactions to painful pictures. So in order to test whether exposure to violent video games may desensitize players of violent video games, I used painful pictures. So pictures in painful and non-painful conditions, as you can see here. So this pilot study included almost 60 adult participants, and I found in this study that in the within design, that participants who played a violent video game versus condition before playing violent video game, I found in this comparison before and after a short-term desensitization, meaning that people who played a violent video game were less sensitive to painful pictures after playing a violent video game. However, it was only found for people without previous exposure to violent video games. And in contrast, I found the habitual desensitization before playing a violent video game in participants with high exposure to violent video games, like in everyday life. So I know it's complex and perhaps difficult to comprehend. So for now, because it was three-way interaction, but I hope it will be just helpful to understand what I was trying to do in my main study. So basically, in my main study, I was thinking perhaps I can check whether the same results could be observed in adolescent players of violent video games. So it was, I would say, almost a direct replication, and the main difference was the age of participants, and there were also some small differences in the EEG sets and in the stimuli used. So Also, the background for my preregistration was the meta-analysis on the topic. So I was lucky that I just found it before I was registering my study. It really helped me to make sure what I have to do in order to analyze my data and how to avoid some typical problems in the empathy for pain research. So one of the problems that was highlighted in this meta-analysis that only about 50% of studies on the topic reported the average number of the epoch that were removed, so the number of the trials that were removed, which is not great. So you should always report how many. If you reject any trials, you should say how many you rejected. And also the key thing about the EEG or ERP studies is that you should be able to justify localizations of the electrodes. And this was done only by 40% of the studies. And with this knowledge, I knew that what I should do better. And I would like to very quickly go through my pre-registration. This pre-registration is also available online, so you can read it more in detail online. And here I would just discuss the 8 questions that you should fill in order to pre-register your study as predicted. So first question asks you whether you have started data collection. So usually you pre-register your studies before data collection. So then you should say, no, we have not collected any data. However, sometimes it could be more complex. It was also in my case. So this is an important tip. So if you are pre-registering your studies as predicted, you need to make sure that your co-authors accept the registration within 1 week. Otherwise, it would you would need to submit a new registration. So it's wise to advise your co-authors to check their spam folder. Otherwise, yeah, you just need to repeat your pre-registration, and that was exactly the case for my study. So I just explained that in detail in the question 8, that, okay, we started data collection, but we did not look at the data, we have not analyzed them. And that I also said that we just made a mistake here in terms of somebody forgot to accept it. So still we think that this is a valid registration. So this is the tip for you. Remember to inform your co-authors to check their spam folder. The second thing is to define the main research questions and the main hypothesis to be tested. So it should be detailed, and as I said before, it cannot be ambiguous, it cannot be interpreted in a different way. So you have to be very specific, just the same way as you state your hypothesis in the final manuscript. In the third question, you need to describe the key dependent variables and how they will be measured. So in this case, I defined my main dependent variable and also the additional dependent variable. So the main dependent variable were the ERP responses, especially the P300 and the LPP amplitudes for the painful pictures. And I also defined the time windows that I expect them to observe between the P300 between 300, 400 milliseconds, and LPP, the late positive potential, around 400, 800 milliseconds. And I also registered that I plan to observe those 2 components in the central parietal sides, so of the electrodes. And additional dependent variable was the pain rating, which was just done by the participants. It was subjective pain rating, so it was not brain data. In the fourth question, you have to define the conditions that the participants are assigned to. So you have to say whether it's a correlational study, experimental, or between, within. So you need to define all the factors that you plan to use, and also how many levels they have. So in here, I defined my between factors, within factors, and this can also match the data analysis plan, which is the next question. So in the fifth question, you have to say how you plan to examine your research questions and hypotheses. So I make the distinction here between the ERP data. So I said what will be my main analysis. So here I said it's ANOVA with within factors. But I also said that I will apply an additional analysis to explore the interaction effects in more detail. And additionally, I also planned to test behavioral pain ratings, so the second type of the data with a similar analysis. So here I also defined the main analysis and the additional analysis. So I think that this is quite extensive description, and it was good enough to. And it was also very helpful for me when I collected the data, so I exactly knew how I have to analyze them. In the sixth question, you have to describe the outliers and how they have to be defined and handled. So why do you plan to exclude some observations? So in terms of participants, I think I did it quite correctly. I said who is going to be my participant and who is not going to be my participant. And then in terms of the artifacts, I only said that I would exclude participants who will have less than 24 trials, because, well, this should 24 trials are needed to make the ERP reliable. So this is the post-factum note that I added here. So probably if I knew all the guidelines for the pre-registration before, I should have include here more details about the pre-processing and artifact rejection criteria, which are of course now in my manuscript. However, I knew some of those criteria before I ran my study based on my pilot. So it would be wise to include them here too. Well, but on the other hand, the as predicted is, as I said, quite, it allows quite short registration. So perhaps if you consider to include all the pre-processing steps, you could use rather open science framework. I also included here information that the time windows were already mentioned in question 3, and also that in the question 8, which is the following question, you can also say more about the pre-processing or the artifact rejection. So what you can do also in the situation like I am now, so I know all what I should have done better. So I would just make my detailed pre-processing pipeline available upon publication. So anyone who would like to check my pre-processing can do it without, I hope, additional questions, that it should be straightforward and detailed. In the 7th question, you have to say how many participants you're going to collect and how you're going to determine your sample size. So here it's really good to base your sample size on an a priori power analysis. However, for some studies, it's very difficult to do it, especially if there is not enough research in a similar design. So therefore, I here said that I'm going to collect at least 46 participants, which is based on the previous studies on the topic, but also based on the previous studies on empathy for pain. This is based on the meta-analysis. And the aimed sample is 60. So for the current study, as far as I remember, I collected 56 participants. So I still fit in the in this estimation between 46 and 60 participants. And then in the last question, you can actually register additional analysis or some variables that you would like to include for exploration, or perhaps something unusual that can happen, but that you can already predict. So I here registered only the variables that I used for individual differences. I also registered my preliminary analysis and also exploratory analysis. And I only mentioned here in one very short sentence the preprocessing, how it will be done, so based on the meta-analysis, but indeed I could have done here better and specify the preprocessing steps and the criteria. So this is my, I would say, take-home message from this session. That's, well, it's very important to registrants share preprocessing code if you don't include it in the pre-registration, because, for example, you didn't know at the time how to do it. But it's also important to plan your study well, and it's advisable to run a pilot. Well, if you're planning to apply some difficult, complex data, then it's best to really look at different meta-analysis focused on the method that you plan to use to see how those studies could be pre-processed and to avoid different difficulties. And finally, you can consider some algorithm that could help you pre-process the data to limit some subjective decisions. So depending on the type of the data you use, there are different available automatized algorithms. For example, here I just added an example of Adam toolbooks that could help you to pre-process ERP data. Yeah, thank you for your attention, and the slides that I just showed you are available at the OSF. And thank you, and I'm looking forward to your questions.